Welcome, everyone. Thank you for joining us for today's webinar, The Future of the Cannabis Supply Chain, presented by Benjamin Chiarelli, founder and chief executive officer, Cellibre. My name is Xavier Gutierrez, and I will be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Benjamin Chiarelli. I will now turn the presentation over to him.
Hi, everybody. Great to be with you here today and appreciate you taking the time to hear about the cannabis supply chain. So before we get into exactly what the talk is today, it's a pretty exciting time to be alive. It's not very often that an industry comes online out of prohibition or otherwise that is this massive and this large, and does so in parallel with a bunch of really cool tech getting invented and implemented across industries. And this table here represents a bunch of the technologies that we see being employed across a bunch of industries today. And I'm here to tell you that the technology that is going to be most consequential, not only for cannabis, but globally, is actually this desk plant. And more specifically, the cells within that plant. Biology is nothing short of the world's most sophisticated manufacturing technology. Every cell on the planet has machines, and those machines are instructed by DNA code to take nutrients and turn them into stuff, and they can do so sustainably and at scale. But why does this matter? It matters because first, cells copy themselves. This means once you have a cell that can make a product, it duplicates itself. The way to think about this is if you're making an iPhone and you dump some water on the iPhone and the iPhone just replicated itself. That's what we're talking about here. You have a cell that makes a product and then it makes copies of itself over and over and over again. Cells also repair themselves. So when cells are manufacturing products, if something goes wrong, they have internal mechanisms to go ahead and fix those issues. This means less downtime, lower maintenance costs, and more continuous production. Cells are also extremely precise. If you think about why cells are manufacturing products, for the most part, they're doing it to survive. And when you're doing something for survival, you want to be very, very precise. So when a cell is making a product, it does so with really, really extreme accuracy. And lastly, cells are a sustainable way to produce products. By definition, they're living organisms. So if they created toxic byproducts, they would kill themselves and stop producing those toxic byproducts. So by definition, they are sustainable. Where is this very, very apparent as cells as a manufacturing technology? Well, believe it or not, cannabis is actually a great example of this. If you go to the 30,000 foot level and say, what is cannabis actually doing and what are those cells doing? It's taking nutrients from the soil, water, and sunlight, and simultaneously manufacturing over 400 chemicals, 100 of which are bioactive. Think about that. If you ask Pfizer or Dow DuPont or Archer's Daniel Midland or any of these big ingredient pharmaceutical or chemical producers to do that type of production with accuracy and consistency, they wouldn't know where to begin with their technologies. But biology can do it simply, easily, and reproducibly. So what we're going to be talking about today in the cannabis supply chain is actually not cannabis or the plant itself and what's known as flower products or the actual plant biomass. What we're going to be talking about is changing the way that we produce the extracted materials, or more specifically, the bioactive materials known as cannabinoids. It's maybe a pretty controversial statement, but cannabis is not medicine. What's actually medicine are these small bioactive molecules found within the plant known as cannabinoids. The most popular and well-known of those is CBD and THC. CBD is the non-psychoactive molecule that is used in a bunch of different products and was recently approved by GW Pharma for the treatment of epileptic seizures. THC is the psychoactive component of the plant, the part that causes the euphoric effect or what's known as the high. So before we get into how Cellibre is going to hack this supply chain, let's first talk about how these extracts are made today. So if you want to make a cannabis extract, you first start with your input. And in this case, the input is a plant. There are a lot of people doing things around genetics of the plant and breeding the plant to make the concentration of those chemicals hit certain specifications. So in other words, if you want more THC, you breed or you genetically modify or you clone a plant that makes high concentrations of THC. Once you have your input or your seeds or your plant that you want to start with, you then grow those plants up. This is usually a 3 to 6 month procedure like you would any plant. You grow it in an agricultural field. And then you go ahead and you cultivate those plants and you take and you harvest them. Once you harvest the plants, you then have to extract the cannabinoids from them. And this is done a variety of methods. The most sophisticated of which is probably supercritical CO2, but there are a bunch of different technologies being developed for extractions. And what you end up extracting is a collection of chemicals. Remember, I said this plant makes over 400 chemicals simultaneously. So when you extract those chemicals, you extract whatever the plant decided to make there. There are a bunch of different variables to control the end result, but what you end up with is a collection of kind of those 400 chemicals in some combination and concentrations of each. If you want to get down to the isolate form, and by isolate form, I mean just a single molecule. So let's say you just want CBD. You start with an input of a plant, usually for CBD, it's hemp, not cannabis, that has high concentrations of CBD. You go through this entire process. And then at the end, when you're trying to get down to just CBD and you're doing your additional purification steps to do so, you have less work to do. If you wanted to make THC, you actually have to start this entire process back over at the beginning and start with an input that has a high concentration of THC. Unfortunately today, getting any of the molecules below that in their isolate form or in their specific ingredient form is almost impossible. And for those where it is possible, it's prohibitively expensive, very, very expensive. And that's just the nature of the fact that the plant makes those molecules in very, very low quantities. So remember back at the beginning of the presentation, I said, biology is nothing short of the world's most sophisticated manufacturing technology. So what if I told you that we could treat cells like that? We could treat them like factories. Remember, every cell has machines. Those machines are instructed by DNA code to turn nutrients into stuff. Well, we now do have that ability. We can actually go into cells, pop out the machinery that they have naturally, put in new machinery, and rewrite their DNA code to turn them into manufacturing facilities for almost a limitless number of products, from textiles to medicines and beyond. One example of this that will probably resonate with people and hit home is insulin production. If you know anybody that's diabetic and has taken insulin, we used to get insulin from a very unsustainable, scalable source, and that is the pig pancreas. We used to actually take insulin from a pig pancreas. Today, we don't use pigs at all. Pigs have been completely removed from the supply chain, and that's because a very smart scientist at Eli Lilly said, well, wait a second, I could change the machinery and reprogram an E. coli cell to make the same exact insulin that we're getting from a pig pancreas. And today, that is how we make our medicines. So what if we took this same concept and we did it in cannabis? Well, it turns out we can. And what we're doing at Cellibre is we have identified 3 different cell lines that we can actually take the machinery, pop it out, pop in new machinery, and use those cells to, in a fermentation process like beer, make cannabis extracts. Here's how this works. I don't want to get too deep into the science, but what you see on the slide now is actually the representation of a cell. And this shows the blueprint for what we are going to engineer into our 3 different cells to start producing cannabis extracts. The input here, instead of plants and having to grow those plants over time, is sugar water. The cell will consume that sugar water and metabolize it into these different pathways, starting with acetyl-CoA. Acetyl-CoA will then go down 2 different paths to eventually produce GPP and alevitolic acid. Those 2 chemicals will then be combined to create CBGA. And from CBGA, we can then create all of the cannabinoids, CBC, CBD, THC, and we will have a cell that produces those. This is how we do the engineering. There are about 17, potentially more, different chemical reactions that we have to engineer in here. Each one of those chemical reactions done with an enzyme or a machine. And eventually we will be able to go from these very cheap inputs of sugar water and produce these molecules. So how will we actually produce it? Great, Ben, you have a cell that you've changed machinery and rewritten DNA code for. How does this actually work? Well, it works a lot like a brewery. Any brewery that you've ever been in. So once we have these cells that make CBD, our inputs, instead of plants, become our cells put into a fermentation tank and fed sugar water. We then ferment those like you would beer, but instead of beer, what is produced is what we've programmed those cells to make, in this case, CBD. What if we want to make THC? Do we run into the same problems that we ran into with agriculture? The answer to that is no. We can use the same equipment, the same downstream processing. We just change the inputs. And actually, we only change one of the inputs. Instead of using our CBD cells, we would use our THC cells, feed them sugar water, ferment them, and you're off to the races, making pure, natural THC. One of the 2 big pushbacks I get on this from a lot of people in industry are, one, people will immediately say, well, Ben, you're making synthetic THC. That is not the case. The way to think about this is that we are making the same natural product, just using a different cell factory. Instead of a slow growth, very complicated, very variable cell like a cannabis plant, we are using very simplified programmed cells like yeast or other types of cell lines. The second one I get is, well, Ben, is it GMO because you're doing gene editing in these cells? Well, remember, what we're making is a chemical. It's CBD, it's THC, it's CBN, it's CBC. Those are chemicals. Chemicals don't have DNA. So by definition, something that doesn't have DNA cannot be genetically modified. What we're genetically modifying is the manufacturing facility, not the actual consumer product. So that's the way to think about those 2 things in the context of the broader cannabis space. So what are the implications of this for the industry? Well, once we have this technology working and it's commercialized, there are a couple really clear advantages. First is the sustainability of production. If you think about what's going on today in cannabis, and there was actually a really interesting study done in 2017 about energy use as an example. If you were to take a 5 million square foot greenhouse and use 100% of that flower for extract, when it comes to scaling our technology, we would be able to do that same volume at in and around 20,000 square feet. This is a really good proxy for resource usage and sustainability. That 2017 study I mentioned actually took a look at the energy use just as one variable in the cannabis industry. And what they found is that in 2016, the US cannabis industry used $6 billion of energy. Just to put that in context, we're talking about an industry that's under prohibition, has not scaled, is done state to state, and really doesn't have any volume compared to what the ultimate market will be. Also, to put that number in perspective, the entire pharmaceutical industry in the US used just over $1 billion of energy. So cannabis, before it even scales, before prohibition has been ended, before it goes global, is already 6x the US pharmaceutical industry in energy usage. We think this is a really, really important point. I think today, when you're dealing with consumer products, medicines, really any product, you not only need to think about the quality of the product and the product market fit, but you also need to think about, when this scales, are we actually being harmful or helpful to the planet? So I think what we're trying to do here is say, look, there's a better, more sustainable way to make these products in the long run, and quite frankly, in the short run as well. The next one is an economic question. The plant, and the beauty of the plant is that it has this really elegant ability to manufacture 400 chemicals simultaneously. But because it has 400 chemicals in it, the concentrations of those chemicals differ. And the concentration of those chemicals is ultimately what drives the plant economics. So we have a graph here, and to give kudos, I stole this graph from Michael at Kronos and Jason Kelly at Ginkgo. They gave this in their speech. They also are working on this science. And what this graph really shows is the cost of a purified cannabinoid per kilogram on the y-axis, and on the x-axis is the cannabinoid content by percent biomass. So how much of the total plant biomass is actually cannabinoid. And what you see here is that for things like THC and CBD, which are kind of on the long tail here of percent cannabinoid content by biomass, you're at around $7,500 to $15,000 in cost to actually extract these chemicals from the plant. But if you want any other plant, that cost starts going up exponentially. Are there other solutions here to potentially breed plants to make those other concentrations in higher quantities? Absolutely. But not to get technical with you, but plant biology is pretty gross. It's very, very difficult to get to work. And actually, THC is a great example of that in cannabis. People have been breeding and genetically modifying cannabis for 30 years to get it THC concentrations higher. And I think the largest I've seen as far as THC percentage of total cannabinoid is in kind of the 30 to 35% range. So thinking about doing that for all 100 bioactive molecules would be a really, really long time. And again, this is just one of the variables. So what do economics look like for our technology? Well, for cellular ag, if you're using traditional organisms, and what I mean by traditional organisms is if you look up this science at all or you talk to anybody that's doing it, they're probably using one of two critters. They're using either E. coli or yeast, and specifically baker's yeast, or what's known as Saccharomyces cerevisiae. The reason for that is anybody with a master's or PhD has used those cells pretty much their entire career, from academia to pharma to industrial applications, et cetera. They use those organisms for good reasons. They have very, very good qualities. They grow very, very fast. And maybe more importantly, we understand them. We know their genetics. We know how they act in fermentation. We know how to manipulate them. Cellibre takes a fundamentally different approach. And it's an approach that is really applicable here for cannabinoids. And that is, we use non-traditional organisms. And the reason that we do that is, instead of trying to take E. coli and yeast and bang those square pegs into a round hole to get them to produce the product we care about, we say, where is there a cell in nature that's industrializable where we're closer to the endpoint? So where using yeast as an example for cannabinoid production might be like using a Ford F-150 factory for producing iPhones. Could you do it? Yep. But you're probably going to have to switch out a lot of machines and rewrite a lot of code and do a lot of retrofitting. And eventually you'll get there and it'll work. What we do at Cellibre is we kind of flip that whole paradigm on its head, and we say, well, what if we could find something that's closer to the end? What if we could find that Samsung Galaxy factory that has a lot of the components and infrastructure that we already need to make our iPhones, and we're just making small tweaks on the edge? Our approach has some disadvantages. The disadvantages are not many people understand these organisms, so actually scaling them isn't as straightforward as it would be with yeast or E. coli. And in addition, working in them isn't the same as yeast and E. coli. The hammer that works in yeast does not work in some of the cells that we're talking about. The way the analogy I use, and you'll see me use a lot of car analogies, so I apologize for that, is it's kind of like if you're using yeast, you're working on the Ford cars. If you're working in some of our organisms, you're working in a Bugatti. It's a very, very special skill set where only a handful of people on the planet, quite frankly, know how to operate it and know how to work on those cars. Better to be lucky than good. We have a scientific team that's been doing this for decades and really understands the organisms that we're going to be working in. As a result of using those organisms, we have some pretty distinct advantages. One is on the IP front, but the other one is just on the economics and the quality that you'll get on the product that you care about at the end. When you're trying to manipulate a cell, you have to remember that a cell, while I'm telling you it's a manufacturing facility, it's not that simple. It's a living organism. And those living organisms have evolved over millions of years to do specific functions. And when cells or living organisms evolve to do functions, they do so for survival. So changing those functions can be problematic. Nature tends to, when you change it, come back and try and kick you in the hind end. So those are all things that you have to consider when you're picking your ultimate organism. And the fact that we're picking organisms that already have the chemistry and biology that we care about to make cannabinoids, that means we have to make less changes to those organisms who have evolved over millions of years. So that provides us a time advantage and also de-risks the project and the program quite a bit. The next one is consistency, and I want to thank Mark Lewis and the team at NAPRO for sharing this data with me. You'll hear a lot of people in cannabis say, well, we have the best genetics and our genetics allow us to keep this plant consistent and we can make a consistent product time and time again. That's not necessarily true. Genetics make up a very, very small portion, I would say probably 10 to 30% of what your ultimate chemical composition is within that flower. Much more important is your environmental impact. So things like what is your heat, what is your temperature, what is your humidity, et cetera, et cetera, et cetera. By the way, that's the reason that you see such high energy use in the cannabis industry is it's really trying to control for that environment. But let me give you one example that none of that will be able to control for. You've probably heard a lot about the human microbiome. That is your stomach and all the gut bacteria that you have in your stomach and how that impacts your health, things like probiotics, et cetera, et cetera. Well, believe it or not, plants are no different. We actually worked for a long time with a small ag company called Monsanto, and they did a lot of great research around the plant microbiome. So in the roots of the plant in the soil, there are actually big collections of microbes that are known as the plant microbiome. And that microbiome actually affects that plant quite significantly, from growth properties to composition, et cetera, et cetera, et cetera. I don't know anybody in the cannabis industry that at scale is trying to control the plant microbiome. And I think when you're dealing with products that have psychoactive or potentially efficacy implications, consistency is really, really important, and controlling a plant is going to be difficult. And this graph here from Mark and team actually highlights that pretty well. What they did was they took an OG Kush plant with the same genotype, and over various batches, they took that same genetics, that same plant, and grew it under a variety of conditions. Organic growth conditions with a biostimulant, just organic with no stimulant, and kind of conventional methodologies using the stimulant. And what you see here is the percent weight of the cannabinoid concentration varies pretty significantly. And then even within that cannabinoid concentration, you have variations in which cannabinoids make up those percentages. And this is just one example in one facility of one genotype. So when you see products out in the marketplace that are labeled as for instance, OG Kush or Girl Scout cookies, all of those different strains, even though they're named the same thing and they have the same genetics, you're not actually consuming the same product. And this highlights one of the issues with consistency. When you're using our technology, what you end up with is a pure cannabinoid isolate. So all we are producing is one cannabinoid at a time. And this production is controlled in that its fermentation, it's GMP, and it's scalable like a bunch of pharmaceutical medicines are made today. About 50% of the world's medicines are using this type of technology. And what we are able to do is not only produce the major cannabinoids like CBD or THC, but we unlock all 100 bioactive ingredients. We can make every single one of them at the same cost, at the same scale, at the same purity. And this does a couple of things. One, it unlocks all of those molecules for study, but two, it also allows you to do true formulations at scale. So let's go back to an example of an OG Kush genetic strain that we do an extract from, and you want an extract that makes up the same exact concentrations of that. What we will be able to do is actually take all 100 cannabinoids plus the terpenes and formulate those into a concentration where they're the exact same and make that product the same way, time after time, year after year, in product after product. So it really does truly provide a scalable consistency across a variety of different areas. All of these factors kind of have different weights depending on your ultimate use case. So there are a couple different areas where cannabinoids and cannabis extracts can be used. The first is the regulated therapeutics market. The leader here is a company called GW Pharma that's both out of the UK and out of the San Diego area. They got the first FDA-approved natural product, cannabis product, through the FDA here recently. And when you're talking about human therapeutics where you've actually taken something through the FDA for a specific indication and you've done those clinical trials, cost becomes actually much less important. They're able to charge a premium for these products ultimately. What does matter is scalability to a point. I try to use GW Pharma here. If it's a smaller condition, obviously scalability doesn't matter. If it's a large condition where there are a lot of addressable, the addressable market is big because the patient population is big, scalability obviously comes more important. But what is ultimately the most important in the pharmaceutical realm is this idea of consistency. Can I have the same product every single time? Because we're dealing potentially with life-threatening diseases here, and these are being used as treatments for those. So having a consistent product that we can take is really, really important. The next market that we can sell into is our cute little cats and dogs and animal health and kind of consumer animal products. Believe it or not, there are some cannabinoids here that are very, very interesting. If you think about what the plan is, it's one big anti-inflammatory. So we've been in discussions with multiple animal health companies about, for instance, helping to treat cats and dogs that have inflammation issues or arthritis issues with certain cannabinoid formulations. Cost a little bit more important here. People don't tend to spend as much money on the dog and cat as they do on the kids, but still they will spend money, so cost isn't the utmost important. Scalability isn't too important here either. These markets aren't as big as human therapeutic markets and human consumer markets, but again, that will depend on the indication. But again, just like human therapeutics, consistency is going to be important here. And I'll give you one great example. There are some cannabinoids that some species of animal just don't metabolize properly, meaning once they consume them, they actually can become toxic to those animals. So if you are using a cannabis extract product today, you should be very, very sure that what you're giving them is, in fact, what is on the label, number one. And number two, you should really understand your sources because it could be detrimental to your little critters. Next, in animal health, there's also the consumer animal health market. We've been talking to some of the pet food manufacturers about potentially cannabinoid formulations into pet food. Here, these are more commodity industries, so your cost is going to be much, much more important. So, going from tens of thousands to hundreds of thousands a kilogram down to, for us, we think ultimately sub $500 a kilogram for these ingredients will be important. These are also much bigger markets. Your dogs eat a lot more food than they do take medicine, so scalability is important. Consistency, not as important here, but again, it's still the purity piece of it is important, but the consistency of the product isn't necessarily as critical as it is in those other two areas, which is why we labeled it as such. And then the last area of cannabis, which is the one that everybody thinks of, is these consumer markets that have been coming online in Canada and in some states here domestically in the US. Here, cost is critical. You are not going to be able to make formulations with specific cannabinoids for hundreds of thousands of dollars into a consumer market. Even CBD products are probably too expensive to get widespread adoption today. These markets are massive. We actually met with one of the 3 largest beer producers in the world and sat down with their scientific leadership. And we did so in one of their facilities that makes over 500 cans of beer a day. So when you're thinking about that kind of scale, consistency and scalability are ultra critical. And again, when you're dealing with human consumption of anything, consistency is always going to be very, very important. Maybe not as important as when you're dealing with life-threatening conditions, but still very, very important nonetheless. So that's why all of these things are very critical. All of the kind of check the boxes on our technology and why we found that this was just a perfect application of this science. And given all of the uses of these cannabinoids and all of the good that they can do for medicine and consumer products more broadly, we're very excited to kind of unlock the currently trapped potential of the plant and the medicines that the plant makes. So with that, I will end the discussion. Pretty picture of me with my email address there. Look forward to answering any questions that you have, and thank you so much for the time today.
Thank you, Mr. Chiarelli, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through June of 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.